Good morning, everyone. It is 10 o'clock Eastern Time. Good morning, good afternoon, everyone around the world listening to us for our final Going Organic webinar series. Today is March 6, 2026, end of the semester. We scheduled this last webinar for giving update from the OREI project as well as the research that researchers conducting using OREI program. My name is Dil Tavaraja. I'm a professor in the Pulse Quality and Nutritional Breeding Program, Plant and Environment Sciences, Clemson University, South Carolina. Our program is funded by the NIFA OREI and USDA ARS, South Carolina Department of Agriculture, and the Global Food Shark. Today, it's very exciting because we are finishing up our OREI presentations, highlighting the research actually conducting these young scientists in my program. The first speaker is Nathan Windsor, one of a very enthusiastic future pulse breeder. He's a he's a PES graduate from Clemson University with the horticulture concentration and plant and environment science concentration as well. And now he's a PhD student in third year of my program. And he is actually developing and looking at the genomic analysis for flavors and also protein content in protein quality of the P genome using the diversity panel. He works with the Spain group in Spain breeder, Dr. Diego Rubiales. And that's our first speaker. And our second speaker is Dr. Carolina Ballén-Taborda. She's wonderful. She is our bioinformatician in our group, and she has a biology degree from Columbia, and she got the PhD in plant breeding, genetics, and genomics from University of Georgia. After that, she joined the Dr. Boyle's program as a postdoctoral fellow on the serial breeding and genomics program. And then she joined my program a year ago as a bioinformatician, and she runs our breed-based bioinformatics programs for all the breeding processes, as well as she managed the chickpea breeding program. And our last speaker is Mr. Mark Dempsey. He is a PhD candidate. He's planning to graduate in August in 3 months. Mark has a BS degree of biology from Appalachian State University and master's in soil microbiology from Miami University. And he has been working more than 10 years in the sustainable agriculture and the organic production. Mark has been tremendous asset to our program for agronomy, soil health, as well as he is planning to be a future lentil breeder. He has been working on lentil genomics and disease resistant, as well as some of the nutritional traits. So you will hear from his research today about pathology work we done in our lab. With that, I'm going to ask Nathan Windsor to start the presentation. Thank you so much.
Okay, thank you very much, Dr. Dill. Make sure you guys can hear me and see everything all right.
Yes, yes.
All right, good deal. Well, I know that we don't have a whole lot of time, so I'm just going to go ahead and jump right into it. So today I'm going to talk a little bit about some of the phenotypic and genotypic insights that we've seen from a global P diversity panel. As Dr. Dill mentioned, I'm Nathan Windsor. I'm a PhD student here at Clemson working in the pulse breeding lab. Okay, so just for a quick outline, today we're going to go over the background, justification, hypothesis, and objectives, materials methods, experimental design, as well as our process for collecting data, go over the results quickly, a brief discussion, and then a conclusion of references. So for a bit of brief background, pea is one of the oldest domesticated crops. It originated in the Mediterranean and Middle East region. It is a nutritionally important crop for both humans and for animals, and it's very high in protein, low in fat, and offers just an overall very well-balanced nutritional profile. They're a cool season legume traditionally grown as a spring crop in cooler climates, which for those of you that have been to South Carolina and spent any time here, you'll know that we are not traditionally a cooler climate, much more heat and a lot more humidity as well. So for a bit of genetic background, peas are diploid with 7 pairs of homologous chromosomes. You can see they have a almost 4.5 genome, and the P reference genome published in 2019 has paved the way for genetic studies in P, which has allowed for a lot more work to be done and really opened the door for more advancements and more studies. So just as a really quick justification for why P's, why South Carolina, and just as a reminder for those of you that don't know or maybe you've forgotten, Clemson is a land grant school here in the US, meaning that it is required to have an extension program that goes out to serve the community and farmers of South Carolina, which is something that I am quite passionate about. I think it's really important that the school serves the state and helps meet the needs of the community, again, both for growers and just consumers here in the state. As you see, we've got a bunch of different sites all over the state that help to really get into the communities there and design, solutions to fit very specific problems. So again, that's something I'm passionate about and think that Clemson does really well. So with that, the reason that we work with peas and the reason I work with them is that they can provide an additional source of revenue for growers without disrupting the typical cropping schedule that you see here in South Carolina. It can be grown here as more of a winter crop, which offers a high value organic production, additive for farmers that might not have been able to grow anything in that season otherwise. And because they are nitrogen fixing, this improves the soil quality and reduces the need for synthetic fertilizer in subsequent crops, again, cutting down on cost and offering a more sustainable solution for long-term farming. Some of the challenges that we're facing with that is that, again, as I mentioned, South Carolina is a much warmer climate, so it wasn't exactly plug and play. Lots of breeding efforts have been ongoing to really figure out how to make a pea that can grow well in South Carolina and survive that higher heat pressure and higher humidity. And then because of this, again, like I mentioned, not a very long cold window here, so the pea needs to grow and fit into a very tight window, get in and out of the field pretty quickly so that the next crop can go in. But these are all things that we've seen in previous work that can be done, and we're very confident in our abilities to make a nice pea variety moving forward. So some quick hypotheses and objectives for my particular study. The first is that significant SNPs for agronomic nutritional traits in the global pea diversity panel can be identified and used to improve and accelerate breeding efforts. The objectives here are to phenotype and describe the agronomic nutritional traits of the GPDP, the global pea diversity panel, as well as identify significant SNPs in the GPDP via a genome-wide association study. And then finally, I'll get to this a little bit more by the end. We're looking to identify and map quantitative trait loci within the pea genome, and that work is ongoing, but we're still growing out this panel, still adding more data, and that final objective will only get improved more as we move forward. So I've mentioned this global pea diversity panel a couple times now. So just to get a little bit deeper into what that actually is, this is a panel that's been assembled using material from Spain's Institute for Sustainable Agriculture, as well as selected lines from the USDA's pea single plant collection. And so this has left us with 364 unique sessions, 325 of those coming from IAS, 39 from the PSP. And then here's just a couple of quick graphs highlighting the diversity of material that we have and also the diversity of, countries represented in this panel. So it covers 12 different species and subspecies from a lot of different countries all over the world. So I feel, very confident in my ability to call it a global pea diversity panel because it truly is a global representation of what we have. And as you can see too, just for fun, we have lots of different flower colors and leaf types within the panel. So as you walk through the field, you get to see, white leaves, purple leaves, flowers, as well as, leafless, semi-leafless, as well as syndrome varieties. So very diverse, really fun panel to work with. And then just for a little bit of heads up as we move to the next few slides, this is the genomic pipeline that we've been working through. So as you can see, we plant everything in the field trials. From there, we have taken our phenotypic data, we've gathered genotypic data, and from there, you can then perform your quality control, data preparation, all of that. Then you can actually perform your analysis and then interpret and validate results. So you'll see this again, but that just kind of gives you a roadmap of what we're going to be looking at in the next few slides. So here's the field trials. Here's my experimental design. We've grown it under organic field conditions at several different locations over several different years. So it's been grown out, since 2023, pretty much every year since. It's got 2 reps within it, and you can see our 2 locations here. The darker star on the top of the state, that's going to be, very close to Clemson, And then that is our Clemson location. And then the middle state is going to be Pelion, South Carolina. So very different soils. It's more of a sandy loam in Pelion, whereas Clemson is going to have that red clay, hold moisture a whole lot more, and just provide a whole different, array of challenges. You can see here's a little bit of what the field study looks like. You can see that sandy soil there. That's going to be at Pelion. And you can see the little peas coming up, looking overall pretty good. And so just to get a little bit more in-depth into the field trial design, they're planted according to an alpha lattice design that's been blocked for maturity. That early block is going to be, or block 1 is going to be for the early maturing varieties, and block 2 is going to be for the later maturing varieties. And within this, we have 6 checks, which are listed there, just to make sure that what we're seeing is consistent across years and have something to kind of compare to. And then this was planted at 120 seeds per plot, 3.5 seeds per foot, 1.5 inches deep. So for data collection, you can see there's another picture of one of our pea fields. That's a very nice looking field. And to take data, what we do is we just walk through it. And, a lot of it is done just visually. And then we also have implemented the use of some drones to get even more data, try to make better, efficient use of our time and our resources. Once we've harvested, we thresh the seeds, grind them in a genogrinder, and then we analyze the samples using an FTMIR spectrometer. This one is the Carey 630. And then so I'm going to highlight really quickly some of the agronomic traits as well as some nutritional values. So first up is the agronomic traits. And you can see this is what we're seeing in the field for just, a lot of different traits really. But I'm going to point out some of the plant height. You see a very wide range in the plants. So some of them are getting pretty tall. Some of them are just barely off the ground, like 63 centimeters to 10. Days to flower, some flower really, really quick at 52 days. Some are taking a lot longer at 97. So the most important, I guess, row I'd point out is just the range there on the bottom. And as you can see, for pretty much every trait, we have a very wide range. Again, just highlighting the diversity that's present in this panel. This shows a significance for all of them. So pretty much everything is significant in some form or fashion. Jumping quickly to the nutritional traits, very similar, chart to what we saw before, where it's just a really wide range for protein, starches, as well as that protein digestibility. Again, what we'd expect from a diversity panel, but still exciting to see it nonetheless. So yeah, moving really quickly through the genomic data, Dr. Diego Rubiales and IAS have sequenced 325 of these lines using diversity raise technology, which is DART-seq. So this is just going to kind of hone in on some of that genomic data collection here. I don't know if y'all can see my mouse, but that first, bit after field trials. And so this is a form of genotyping by sequencing of GBS, and it's also, a form of NGS. Some of the TSP lines are not yet sequenced, but they're currently being genotyped and will be included in all future analysis. That's one of those things that I'm working on right now. So just to quickly show through some of the results, here is the SNP density map. So you can see it's very dense. We have a lot of really good representation there. And then this PCA chart here kind of shows that most of our, I'm blanking on the word right now, sorry, y'all. But yeah, the principal component analysis, a lot of the main reason for this, deviation is due to the material and the species type. But a lot of it, is still up in the air, very likely genetics. So just to move through these really quick, I have a lot of very similar slides, so we'll go pretty fast here. But this is a Manhattan plot, kind of showing that we have identified, possibly some SNPs for plant height. And this is consistent with some previous studies that have found, pretty similar SNPs in this region relating to plant height. Again, days to flower, very similar. What we're seeing is consistent with the literature. That's going to be the same for days to maturity, as well as the average seed weight and yield. So this is really exciting. We've got some really promising results, and we're definitely looking forward to furthering this analysis to confirm what we're finding and just, learn even more. So I know that was really fast. So in a quick discussion, these identified SNPs can be used to make better informed decisions in a breeding program, which leads to improved varieties, accelerated releases, which is just going to be, overall better for, breeders like ourselves, as well as farmers and growers. And then just to touch on this briefly, there's been an additional recombinant inbred line study that's going on to investigate protein concentration in peas. This is going to have a QTL identification and analysis. And then the results of both of these studies are going to be, compared and contrasted to one another and used to corroborate each other. So finally, as a brief conclusion, there appear to be significant SNPs for the following agronomic traits, including plant height, days to flower, days to maturity, as well as the average seed weight and yield. Continued analysis will improve data and results, and the genome-wide association studies will expand to include additional agronomic traits, nutritional traits, and nutritional traits. And I wanted to mention that we are currently, within that 2026 field trial year. So a lot of this data is going to be improved over the course of this summer with, hopefully more promising results later this summer or this fall. And just as a really brief mention, you can see I've got a picture of a dog there. That like my last field season 2025 was one of the weirdest ones I've had all sorts of random issues in the field, such as, trucks driving out over the plots, just having some fun ruining our study, as well as a couple of dogs running through that weren't supposed to be there. So it made for a very interesting field season. And now I've got another dog, which was, a good bit of fun, nothing that we expected to harvest this year. So there's some references, and just wanted to acknowledge the lab team, my committee members, and also our funding and support from the USDA NIFA OREI program in particular. Thank you very much for your attention. Sorry I was moving so fast. And if you have any questions, let me know. I think we're going to do a question and answer period at the very end. So thank you for your time and attention.
So yeah, today I'm going to talk about a genome-wide association study, or GWAS, of protein in pea for biofortification. And so Nathan did a great introduction about the crop, but I would like to emphasize that pea is a very valuable pulse crop, and it has a very dense, nutrient-dense seed composition. And since the focus of the presentation is protein, I want to mention that pea seed has between 20 to 25% protein, and has been reported as high as 33.9%. So this is really, really good. And on top of protein, it has a very other nutritional content, including fiber, starch, protein digestibility, and vitamins, minerals, and carbs, but is low in essential sulfur-containing amino acids. So this is really key. And another thing is protein is very valuable for sustainable agriculture, soil health, and fertility, because it fixes nitrogen, atmospheric nitrogen into the soil. It is very water efficiency, and it requires less inputs. And since the plant-based protein food is in increasing demand in the world, we are exploring pea as an alternative to animal-based foods. And having all these benefits for pea, it has received less attention for breeding, especially in the southeast of the US. So there is a lot of opportunities for genetic improvement of nutritional and quality traits, and also yield and other traits. So the objectives here were to identify regions in the genome where there is association with protein and other nutritional traits. So for that, we wanted to quantify the phenotypic variation in a diversity panel of pea, characterize the population structure, and also find genomic regions associated with the traits. For this, we evaluated a USDA pea single plant plus collection. For short is PSPPC. It contains 267 accessions that come from different parts of the world, from 59 different countries, as you can see here in this map, and mostly they come from India, Ethiopia, and European countries. And we had the previously report developed the genotype for these accessions, and it contains around 54 SNPs. And then this genotype was used to run population structure analysis, to do principal component analysis, and also to understand the genetic diversity by estimating nucleotide diversity, Tashima's D, and heterozygosity. This population or panel, the diversity panel, was evaluated in the inorganic fields in South Carolina. along with two checks, Carver and Inca. It was evaluated in a randomized complete block design with 3 reps, 3 years, and 2 locations for a total of 3 environments. Here you can see the first environment was planted in close to Columbia in Pillion, South Carolina, and the 2 other environments were planted in Clemson in an organic field in 3 different years, these experiments. And all the seeds were harvested, threshed, and grind into powder, and the powder was run through the fluoride transform mid-infrared spectrometer to obtain a spectra. And this spectra was transformed into values using previously trained models, and the data was adjusted for 15% moisture. And these models will give us information about the protein, sulfur-containing amino acid, fiber, starch, and protein digestibility. And with this information, we calculated blobs because we had information from different environments, and we also run correlations between the traits to understand their correlations or their relationships. And if you are familiar with a GWAS analysis, it receives genotype and phenotype. So these were inputs for GAPIP, and we run different statistical models to understand what regions in the genome are controlling these traits. And also, once you identify the location or the significant SNPs, you can calculate linkage disequilibrium blocks using PLINK to identify the whole region controlling the traits. The population structure shows us that this diversity panel has 10 subpopulations. As you can see here, all the vertical lines indicate accessions, and the numbers indicate how many accessions are in each subpopulation. And if you can see here at the right of the screen, there is a subpopulation that has the most admixture with other subpopulation. We also can see that in here with the PCAs, which are explaining 18% of the variability. If you can see here, the green cluster is also kind of in the middle of all the clustering, which means also this is very highly admixture with other clusters. And if you can see here, like less accession with least admixture are kind of like the brown, red, purple, and pink. And finally, the diversity estimates for like pi, Tajima's D, and heterozygosity are telling us, these numbers are telling us that this panel is highly diverse, this is great for breeding. We have so much genetic diversity that we can leverage to improve P, not only for nutritional trait, but also for disease resistance, agronomic yield, anything you can think of. The correlations between traits, here I'm highlighting high positive correlations between protein digestibility, and fiber. I'm highlighting this because this is good, because if I'm improving for higher protein, is it likely that I also would be improving fiber and protein digestibility. This is something that we would like to see to make for their genetic gain in the breeding program. This is a PCA by plot with the phenotypic information. If you focus right here, we also see that these 3 traits are highly correlated. And for example, if we select lines around this cluster, we can use these plants as donors of high protein, high fiber, high protein digestibility, or we can cross it with lines that are low for these traits to create mapping populations for further validating the QTLs. Finally, the GWAS show us that there is 17 significant SNPs. More importantly, we found a location in chromosome 5 that is really important, highly associated with protein and sulfur-containing amino acids. So this region is controlling these two traits. And this region is explaining 18% of the variation. And this is really compelling results. Like we found these across and within environments, and we will use this information to target this region, to bring this region into elite cultivars. Takeaways, this study evaluated a high diversity panel, the PS-PPC, that show a high genetic diversity. We found a chromosome segment or loci chromosome 5 associated with two important traits in P. And the final conclusion is that understanding the genetic basis of nutritional traits, including protein, can facilitate the development of South Carolina adapted superior cultivars that are resilient, high yielding, and biofortified. And also these give us opportunity to expand the potential of P to becoming a plant-based or like a crop for plant-based food systems. And with that, I would like to thank all the team, the Pulse Quality and Nutritional Breeding Program, our collaborators, all the resources that we have at Clemson University, including the supercomputer that we use for our analysis, and of course to the funding agencies for allowing us to do this research. And thank you very much for listening.
Okay, hi everybody. Please let me know if you, for some reason, can't see the shared screen.
Mark, can you please put into the screen mode, presenting mode?
We good there?
No, it's small. We don't see the full slide. Like we see, there you go.
Okay, great. Thank you. Okay, hi everybody. My name is Mark Dempsey, and I've been working on adapting lentil to the Southeastern United States for the past few years. Lentil is not currently grown in the Southeastern U.S. It's currently grown, however, in the Northern Great Plains and Palouse regions, and importantly, there it's grown as a spring and summer crop. However, organic grain growers in the Southeastern U.S. stand to gain from lentil. There's regional demand for organic pulses and a significant price advantage for organics. Lentil in the Southeast could be an easy swap for winter wheat, importantly grown as a winter crop instead of as a spring-summer crop in the northern regions. And it uses the same season and equipment as winter wheat might. Lentil can lengthen crop rotations, potentially reducing disease and insect pressure, and can improve nitrogen management for the subsequent crop. Yet, many adaptation challenges remain, wetter and warmer climate here compared to major production regions, leading to higher disease and weed pressure. There's limited pest control options in organic production. Late spring heat can be problematic, especially during seed development. And with these things in mind, the ideal genotype for the Southeastern U.S. should resist disease pressure, be large stature to compete with weeds, have high yield potential, and maybe mature early to avoid that late spring, early summer heat. To address these challenges, we conducted a 3-year field experiment. The first study within that experiment was agronomic traits for organic production in the Southeast. The objective of this was to identify genotypes with favorable agronomic traits for use as breeding parents in a breeding program downstream. Those favorable agronomic traits include large stature, again, to compete well with weeds, low lodging for good mechanical harvest, high yield, and a low stress response. I put an asterisk next to stress response because we believe that the major cause of stress symptoms observed in our study was due to disease, but the study wasn't really designed to untangle biotic from abiotic stress. We didn't go through the steps of isolating pathogens, inoculating our entire panel with isolates and evaluating them from there. And further, the soil nutrition and moisture or rainfall appeared to be sufficient. So from this, we believe that stress response was primarily caused by disease, but conservatively, I'm just going to refer to this as stress response. And in the second study is genomic regions associated with that stress response, where the objective is to identify genomic regions associated with that stress response to inform future breeding work, ideally to generate some molecular markers that can improve selection efficiency down the road. So For this field experiment, we used 287 or 446 genotypes, depending on the year. And across all years, we used a subset of the LSPD panel, that is the Lentil Single Plant Derived Core Collection panel curated by the USDA. We used 278 genotypes from that across all years, 287 across all years. And then in the third year, we integrated 159 additional genotypes from ICARDA. So there was 129 genotypes from the heat tolerance panel and 30 genotypes from the Global Association Mapping Panel. Again, both of those panels come from ICARDA. We planted these across 5 environments. This was 3 locations across 3 years. So our first location is Rawl Farm, where we grew this population out for 3 years. Rawl Farm is located in the sandhill regions of South Carolina, which is dominated by sand or sandy soils at about 90% sand and a relatively low clay content. In 2023, we planted this at Clemson's Cherry Farm. This is in the upstate of South Carolina, which is in the Piedmont region, and soils are characterized by a higher clay content between 25 and 30%. In 2024, we conducted the study at Clemson's Simpson Farm. The study was conducted using a alpha design with 2 reps per environment and several incomplete blocks per rep, and many check plots within any incomplete block. We use the variety CDC Maxim as a check variety. All plots were established within the first 3 weeks of February across all 5 environments. They were planted on 15-inch or 38-centimeter rows and 130 seeds per meter squared in the first 2 years and 86 seeds per meter squared in the final year. So moving on to the first study, agronomic traits for production in the Southeast. As a reminder, the objective is to identify genotypes with favorable agronomic traits that can be used as breeding parents. To do this, we measured 6 traits in the field. The first is a stress response was scored on a scale from 1 to 5, where 1 is essentially good and 5 is bad. This was done based on the percent of the canopy showing visual symptoms, and that was done about 80 days after planting. Visual symptoms were primarily chlorosis that started at the bottom of the plant and progressed upward. Any signs of foliar diseases or vascular wilt were relatively rare. And the major symptom of stress was chlorosis. Plant height was measured on 3 plants per plot. Canopy cover was measured using fractional green canopy cover from digital images collected approximately 80 days after planting. Lodging was scored on a scale from 0 to 5, where the score of 0 was 0% lodged and the score of 5 was greater than 85% lodged. Yield was measured as total seed weight per plot, and days to maturity is when 90% of pods in a plot were brown. Mixed effects ANOVA was used to test the effects of genotype, environment, and the genotype by environment interaction. Estimated marginal means were then calculated, and in order to identify groups of genotypes with favorable traits, higher cluster analysis was used to separate genotypes into groups based on 25 trait environment combinations. To improve selection of genotypes, filters were then applied using EM means. The first filter I call the better than average filter. I'll explain what that means a little bit better on subsequent slides, but briefly, we identified the average value for a trait for across the population, and then selected the genotypes that were better than that average value for that trait across all traits. But because that didn't capture all of the genotypes that we wanted, particularly in the groups that we felt were the best performers, we narrowed in on some of the top yielders and loosened up the constraints otherwise, and I'll explain that visually better in 2 slides. And all stats were performed using R. A quick side note about disease diagnostics. Disease plants were sampled when symptoms were severe. 3 diseased plants per environment were diagnosed at Montana State's Scudder Diagnostic Lab. Many thanks for their help in this process. And at Clemson's Plant and Pest Diagnostic Clinic. For the results of these diagnoses, root rot was the most common disease observed across the study, and the onset was typically during reproductive stages. Fusarium was the most common pathogen associated with root rot. And unlike root rot, wilt and foliar diseases were relatively rare. This table summarizes the results of these diagnostics, and you can see that Fusarium was observed consistently across all samples, either morphologically or using genetic markers. When genetic markers were used, Fusarium oxysporum was the most common species observed. And there was often, if not always, a oomycete observed in these samples and was identified as Pythium when it was successfully identified. But when it wasn't, it was suspected to be either Pythium or a Phenomyces. Okay, moving on to results from the study. Again, these are going to be results for the cluster analysis, where the goal is to separate genotypes into groups that have favorable traits. So on this graph, each dot represents the value of a genotype. And you can see that along the x-axis, the cluster analysis separated all genotypes into 12 different clusters based on consistent differences across traits and environments. And within this graph, you can see that some clusters are consistently high and some clusters are consistently low for their stress response score. And the same is true for plant height, where some clusters are tall and some clusters are short. And similarly for plant canopy and other traits. And careful investigation of these traits across clusters narrowed in on clusters 7 and 8 as having a low stress response score, being tall with a high canopy cover, intermediate or a widespread for lodging and yield, and a relatively late days to maturity. But notably, the cluster 7 and 8 did not contain the highest yielders among this group. And so cluster 3 was also selected because it's high yielding, but happens to be small in stature and shorter to mature. And together, these 3 clusters represent 80 genotypes that possess favorable traits that could be used in a future breeding program. But I think that we could do better than starting with 80 genotypes and narrow it down a little bit more to really start with the best of the best. Hence, applying filters to really narrow in on genotypes that we believe have the most favorable traits among these. So filter 1, again, is that better than average, where we identify the average value for a trait and select the better genotypes below, in this case, below that average value, or those for plant height that are higher than the average and canopy that are higher than the average, so on and so forth. Apply that as filter 1. And then for filter 2, we're focusing in on the top 10% of yielders because we really want to capture the highest yielding among the bunch. But then we have to loosen up constraints to get the smaller statured but high yielding ones in the door. And from this, we were able to identify 24 genotypes within clusters 3, 7, and 8, and those are shown with either red or blue circles around them. So the outcome of this research is that we identified 24 genotypes with favorable traits that show promise as future breeding parents. Those genotypes are shown here with their cluster numbers in the table. Ongoing research this season, we currently have a study out in the field evaluating these 24 genotypes. It's a more robust study in that it has more reps and includes both resistant and susceptible checks to try to get a better sense of what's going on with disease, resistance, or susceptibility. And importantly, we'll be doing improved disease identification and phenotyping this year. So moving on to study 2, genomic regions associated with stress symptoms. Again, the objective there is to identify those genomic regions associated with stress response to inform future breeding work, again, ideally to generate some molecular markers to improve selection efficiency down the road. So to do this, we use best linear unbiased predictors, or BLUPs, generated from stress scores from the field experiment. Genetic data consisted of genome by sequencing data converted to VCF, then filtered and imputed. And genome-wide association study was performed using GAPA and R. The results identified many significant SNPs. There were 5 significant SNPs across 4 chromosomes, and then 4 genomic regions within chromosomes 4 and 5 that had a high density of significant SNPs. And when examining each SNP on a SNP-by-SNP basis, we were able to identify several significant SNPs that had differences in those stress scores. And by that, I mean, if we take a look at, for example, two SNPs from that band in cluster 4, you can see that the stress response score for the genotypes with the rare allele were on average lower than those with the common allele. And this is also true for one significant SNP in chromosome 7. And just to illustrate that a little bit, there's a picture of a plot from a genotype with the rare allele compared to one with that common allele. Linkage blocks were then identified for these two SNPs from chromosome 4. Again, there are many in that genomic region where you can see that there are several significant SNPs. There's hundreds. And within these linkage blocks, we have yet to finalize the examination of candidate genes within this region. And so we are still conducting this analysis and hope to identify candidate genes within this region that are related to those differences in stress scores observed. So the outcome of this research is to identify significant SNPs where the rare allele has lower stress scores. And ongoing and future research is to investigate those SNPs more carefully on chromosomes 4 and 5, again in that SNP-rich region. And then within linkage blocks, examine candidate genes to try to get a better handle on what might be contributing to the differences in stress scores between the rare and common allele. And then, of course, the next step would be to make some crosses to create a population where we can validate SNPs in a segregating population for this trait. And as a secondary but different goal is to isolate and identify pathogens from South Carolina fields to really try to start answering the question, is the stress response observed due to disease? And so if we were able to successfully isolate pathogens, then we could take that to a greenhouse for a controlled study where we can screen for tolerance and resistance after applying consistent inoculum load to, for example, the full panel, or even just start with our 24 genotypes of interest and see how they perform in response to those pathogens. Here's some references for this talk today, and I'd like to thank our funding sources, my advisor, Dr. Dill, committee members, and the lab team. Thank you, and I think we'll turn this over to questions.
Thank you, Mark. That was wonderful. And everyone, it's great presentation, great research output. Thank you so much for presenting this. I'm going to start the question with Nathan Windsor. So Nathan, so you are the most youngest family member in our group, and what you think of being a breeder, a future breeder, how can take your project to the next level? What would you planning to do the next level of analysis?
Yeah, it's a good question. I think the main thing for me with this project in particular is just going to be continuing the analysis that we've already started, but adding in additional locations, additional site years, and just getting just in greater depth. So I know right now we've got, I mean, I don't want to say preliminary, but just some early data showing that, there are some SNPs there. Some of it's been confirmed in previous literature. Some of it, is looking to be new. So just continuing to dig deeper into that, kind of proving what's there. And then, just increasing the study to contain even more data and just more detail.
Yeah, that's wonderful. Maybe you will find more dogs,
Maybe, we'll see. I can't take any more. The one's crazy.
Exactly. And then there is a question in the Q&A for you. It says, do you consider study the quality traits of the pea at their earlier stage? As we know, peas are consumed mostly for their young pea seeds. What do you want to talk about that? I know it's a little confusing, but what do you think of that?
No, I mean, I think it's definitely something worth considering. For the scope of this particular project, that wasn't exactly our goal from the outset. We were definitely more interested in the protein concentration and protein quality of a dry seed. But I mean, there's definitely a lot of value in looking at the younger qualities of the trait, or the traits of a younger plant. We take data on it as we go. So I'm not harvesting young seeds or anything like that or immature seeds, but I am making note of disease and plant height of some of the younger plants, just keeping an eye on them. I definitely think there would be value in future studies looking into that more, because I think that the genetics are going to be the same. So if we've got some varieties that come out of this panel that would maybe not be the best, for a mature seed, but have really good qualities of a young plant and are really tasty, like absolutely we can get into that.
Yeah. So basically, that is a different breeding objective. So our breeding objective is to make the dry peas, right? So that means we harvest at the dry condition. So the vegetable peas, that's a different objective. So, but Nathan is actually studying the pea diversity panel, which has the vegetable peas as well as the dry peas as well. But it's the difference is the morphology of the plant as well as the pot. So yeah, but that's a good question and a good answer. We could collect it, but since our major objective is a dry seed, so that's why we do that. That's great. So then let's see. All right. So next question is for Carolina. So Carolina, you are handling a large set of large data. So what kind of opportunities we have at the Clemson to do this job easy? And also, what the message that you wanted to give to the breeders?
So the first question, yeah, we have a lot of resources at Clemson, including the supercomputer. It's called Palmetto server, where we run all kind of analysis. We have a large memory, high storage, so we can run everything there, and it's very quick. So we have those resources available, and also the managing team, they train us how to, scripts if you are new with Linux or with R. They provide all the training that we require, and of course we help each other in the team to run all the analysis. So we have a lot of resources at Clemson. And the second question, sorry, I forgot.
How this help to the breeders?
Oh, how it helps to the breeders. So yeah, we can store all the data. We can analyze, we can make plots to help us make decisions, like running, for example, the GWAS very fast to find the genes, run all the genetic analysis, analyze large phenotypic, genotypic data. So it helps a lot to the breeders having these resources available. And we all the time are using these resources. I hope that answered the question.
Yes. And then what is your, okay, now I'm going to connect with the Nathan's question. So the question was like getting the young parts. So, but we are working on dry seed, but it doesn't matter for genomics, right? So can you, if someone wanted to study our panel for young peas, vegetable peas, can they use the same panel? Can they use our data sets? Can you comment on that a little bit?
As far as I understand, this panel is mostly for dry peas. So, and all the data we collected is on the dry peas and the